a freshman at Biophotonics lab at KAIST. Thank you.
Hi everybody, my name is Puya and I'm a master's student at Brain Dynamics Lab, and I'll be the moderator for this seminar. Thank you.
Hi, my name is Youngil Cha, under Professor Kyun Jung's lab, and I'm working in manufacturing the optical color filter array. Thank you.
Hello, I'm the second presenter, Jaehoon Chang, and I'm also in the Biophotonics lab in KAIST. Thank you.
Hello, I'm Jun Tae from the Tripoli lab of Professor Choi Kim. I will introduce you about controllable biosensor today. Thank you.
Hello, I'm Yongyi Chang. I'm pursuing a master's degree in Choi Kim's lab, and my research interest is wireless power transfer. Thanks.
Okay, I think all of our members introduced themselves, so I'm just going to pass the baton to our first presenter, Youngil Cha. So Youngil, why don't you just take it off?
Hi, my name is Youngil Cha, and I'm first the presenter of this topic of biosensor. I will briefly introduce the concept of biosensor and its configuration. So, to be generate, what is the biosensor? So, the figure you're looking at is a pregnancy test kit, one of the most successful biosensor in the world. With all the features of good biosensors, this machine reacts with HCG, the human hormone, in the urine to indicate that you are pregnant. So pregnancy test kit is composed of the sample pad and the reaction line, the test line, and the valid line. The sample line is the sample is the human chorionic gonadotropin, the HCG in urine, and there is the loosely attached enzyme conjugated anti-HCG antibody on the reaction line. The polyclonal anti-HCG antibody on the test line and polyclonal anti-HCG antibody on the valid line. The principle of the pregnancy test is as follows. When the clinical sample with analyte touches the strip, the process through the capillary flow. In the reaction line, HCG binds to loosely attached enzyme conjugated anti-HCG antibody, the through antigen antibody reaction. After that, modified HCG and immobilized HCG antibody are combined in the test line. The final result can be confirmed because the immobilized anti-HCG antibody binding to the antibody is in the valid line. So, like this example of biosensors are the analytical device that convert a biological response into readable the signals. The transducer converts the signal created by the combining the sample analyte as the anticorps, chemical proteins, DNA, etc. with the receptors into an electronic signal. Not only the electronic signal, it can be the readable signal. So as you might think, the various applications have been developed according to the material and structure, operation method, and the fabrication method of receptors and transducers. So there are several types of receptors. First, enzyme-based. Enzymes are the most generally used biological components of biosensor. They have advantages of the right number of the reactions they catalyze, the possibility of detecting a broad range of analytes, and the different transduction principles that can be used to detect analytes of interest. It has been studied a lot throughout the combination with various materials such as the nanoparticles. And second is the antibody antigen-based bioreceptor. The unique capacity of antibodies to bind specifically the analytes of interest is the key factor in their usefulness in immunosensor design. As shown as the figure, there is 4 types of conventional antibody-based methods. In direct assay, the antigen is incubated with assessed amounts of the immobilized antibody, and the interaction is detected. And the second one is the competitive assay format are based on the competition between an analyte derivatives and the analytes in the sample for a limited number of antibody binding sites. In binding inhibition assay, the antibody and the analytes are the first incubated like this. After evaluation, the solution is placed in contact with the immobilized antigen. So as I already mentioned, the pregnancy test kit. In the sandwich assay format, the antigens are incubated with an excess of a primary antibody, and the resulting antigen antibody complex is incubated with a second Warburg antibody, which binds to a second antigenic site. Okay. And other bioreceptors are configured nucleic acid-based and biomimetic and the cell-based. The nucleic acid-based receptors can be either based on complementary base pairing interaction, referred to as genosensor, like this, or specific nucleic acid-based antibody mimics. It can be the certain include of the biomimetic, but it also detects the nucleic acid as it calls aptamer. The nucleic acid-based biosensors is generally used for detecting the RNA and DNA with nanoparticles or fluorescence labeling. And the fourth one is the biomimetic sensor is the artificial receptor. It contains genetically engineered molecules, artificial membrane, and molecularly imprinted polymers. The design and synthesis of artificial receptor systems with antibody-like recognition properties have recently attracted much interest. And the last one is the cell-based. We can use the living cells as the bioreceptors. This type of biosensor has the advantage of more tolerance of environmental changes as the pH or the temperature changes. Single cell can contain all the enzymes and cofactors needed for detection of analytes. So roughly, biosensor can easily be regenerated by letting the cells regrow. And also, but however, has some limitation of the full selectivity. After that, after binding the bioreceptors with analytes, the transducer converts the biosignal to readable signal. The various types of transducer are reported, and I overview the common types of transducer. Only the common types of transducer, and there is the 4 types of transducer as the electrochemical and optical, mass sensitive, and thermometric. widely used for biosensor. Since there is so many principles and contents of transducer, I will briefly explain only the most basic content and examples because it's just briefly overview. So at first, the electrochemical transducer. As shown as the figure, electrochemical biosensors are normally based on enzymatic catalysis of a reaction that produces or consumes electrons. A typical example is an invasive blood glucose sensor. As you all know, the glucose concentration is measured by the measuring electrons related to reduction oxidation reaction like this through the glucose oxidase. In addition, the method using potentiometry, amperometric, and conductance and capacitance are widely used for the electrochemical transducer. And second one is the optical method. The basic object of an optical biosensor is to produce a signal which is the propotentiate to the concentration of measured substance. Measure the absorbance, the reflectance, luminescence, refractive index, the fluorescence, and the light scattering, and peak variation of an object to extract quantitative data. The measurement method using infrared is in the spotlight as a non-invasive biosensor. And third one is the mass-sensitive transducer. Mass-sensitive transducer has very intuitive working principle. It's just to make a scale that is very small enough to measure the molecular weight. The scale is composed as cantilever and nanoparticles. The signal is change of the phase and frequency and amplitude. Mass-sensitive biosensor has the advantage of the quantitative sensitivity. The last type of the transducer is the thermometric. Thermometric method measure the heat generated when the receptors and the analyte bind. One of the most popular ways of thermometric biosensor, the enzyme thermistor is the flow injection calorimetric biosensor that directly detects the heat evolved during enzyme catalyzed reaction. So far, it has been a basic concept for the biosensor. Thanks for listening.
Right, thank you very much. Our next step is going to be Jehan, and he's going to talk about the clinical applications of biosensors.
Okay, hello, I'm the second presenter, Jae-Hong Jung, and now as I told you before, I'm in the biophotonics labs, and my research field is about the MEMS device for the spectroscope. My presentation topic for today is biosensor for clinical application. Here is the content. First, I will present about the working principle of biosensor very briefly, and introduce the importance of the biosensor for the clinical application. Next, I will present some examples to show how biosensor can be applied clinically, such as the glucose meter, biomarker detections, and viral infection diagnosis. Finally, I will finish my presentation by showing some further application. First, as the first presenter explained, the biosensor consists of the 4 parts: the sample, biological elements, transducer, and signal processing. Each part is selected based on the target of detection. The biosensor can specifically detect the target substance in the body, so it can be used in various medical applications such as monitoring and diagnosis. As shown in the market analysis graph above, the biosensor market in Canada is now worth about $1 billion and continues to grow over the year. Especially the market is expected to have a great influence in the clinical field. Blood glucose meter is an example to show the impact of the biosensor in clinical field, which has already been commercialized. In addition, the proportion of the biosensor used for research purposes, such as immunofluorescence analyzer, is also increasing. Such commercialized biosensors are already used in various fields, particularly as the rapid infection diagnosis required in the current pandemic situation. The COVID-19 diagnostic kits with the high accessibility using biosensor have also been commercialized. And this situation will accelerate the growth of the biosensor market. From now on, I will present some examples on how biosensors are currently being used for the clinical application and introduce some recent research. The blood glucose meter is a representative biosensor for the clinical application. Blood glucose monitoring is very important for people suffering the diabetes, and various studies have been conducted for it. The enzyme-based glucose, the enzyme-based glucose meter has been commercialized and used by many patients to measure the concentration of the glucose. It uses the glucose oxidase as the biological element in biosensor. As shown in the figure below, the concentration is measured with the electron generated by the hydrogen peroxide. Hydrogen peroxide as the result of the glucose oxidation. When the blood of a patient is dropped on the strip, the glucose oxidase in the working electrodes reacts with the glucose in the blood and generates the electrons. And it causes a different current depending on the concentration, and the concentration in the blood quantified by measuring the electrical intensity. Other than the monitoring target molecules by previous example, but also the several studies to diagnose with the biosensor by detecting some indicator in the body. And we call this indicator as the biomarkers. Biomarkers are the specific substance such as the proteins and DNA, and used as the indicator for detecting the changes in the body. Biosensor can be used to diagnose by sensing a highly related biomarker to the specific disease. For example, the troponin I is a cardiac and skeletal muscle protein family, and concentration of it in the blood is increased when the cardiocytes are damaged. So it can be worked as the biomarker for the cardiac damage. The right figure shows one of the biosensor, the BioF18, with the silicon nanowire substrate. The antibodies on the substrate bind the troponin I specifically. In this system, when the troponin I is combined with the antibodies, the charge distribution on the surface changes according to the electrical surface potential on the semiconductor, and it changes the amount of the current between the source and the drain electrode. So the concentration of the troponin I can be quantified by the intensity of the current. And this research implements the super sensitive sensor with the 5 picogram per millimeter of the limit of detection by utilizing honeycomb structure silicon substrate. Next example is Alzheimer's disease diagnosis by detecting 4 biomarkers. The amyloid beta peptide and phosphorylate and tau, the total tau are the biomarkers for the Alzheimer's disease, and the concentration of the cerebrospinal fluid changes 10 years prior to the onset of the symptom. In addition, the several research reports that the Alzheimer's disease is related to the concentration of those markers on the plasma. So the biosensor for detecting those markers in the plasma reduces the risk by early diagnosis of the Alzheimer's disease. This biosensor can measure the concentration of them by measuring the change of the electrical resistance when each biomarkers are bound to the antibody immobilized on the substrate. In plasma, these biomarkers are 100 times lower than in cerebrospinal fluid and contains other interfering substance. So sensitive measurements are essential. Therefore, in this study, carbon nanotubes are densely aligned to reduce the conjunction for lowering the electrical resistance so that the sensor reacts sensitively to target binding. Each biomarker's concentration was quantified as the change in the electrical resistance and showed significant difference against the control. Next, the quick and accurate viral infection diagnosis is important in current pandemic situation. Biosensors can be utilized for the viral infection diagnosis. The diagnosis can be divided into the direct method and the indirect method. Gene detections using some techniques such as the PCR or the viral isolation are the accurate but less successful. On the other hand, indirect method which detect the antibodies on the with the compact biosensors can improve the accessibility. As shown in the figure below, when the infection occurs in the body, immunoglobulin M and immunoglobulin G are formed after 3 and 7 days respectively. Therefore, the infection diagnosis can be implemented by judging whether the immunoglobulin M and G or G have formed in the body. Based on this principle, the COVID-19 diagnostic kit was developed to detect the antibodies in the blood. There are 3 lines on the strip, and each line is immobilized with anti-human immunoglobulin M and immunoglobulin G antibodies that bind to the COVID-19 antibodies. The antigen is conjugated with the colloidal gold nanoparticles and treated with the patient's blood on the sample pad. If the COVID-19 immunoglobulin M and G are present in the blood, the antigen-binded antibodies is bound to each line. And this is indicated by the red line on the diagnostic kit. And the red line except the control line means the patient has a risk of the virus infection. This method can only be diagnosed when the antibodies are present in the body, but patient at risk of the infection can quickly diagnose individually without any other specialized techniques such as PCR, so it can prevent the further infection. So far, I have introduced the various biosensors and its example for the medical application. Biosensor can be utilized in the wearable healthcare device as shown in this figure, and by combining various flexible and miniaturized sensors, especially the several non-invasive and minimally invasive glucose sensor to utilize in the wearable device and continuous monitoring have been developed. So finally, I will close my presentation by telling the biosensor can be applied to wearable device for monitoring specific substance inside the body. And thank you for your listening.
Okay, thank you, John. Next up is our first panelist, Yongki Chang, and he's going to talk about field effect transistor-based biosensors. So Yongki, take it up from here.
Okay, I'm Yongyi Chang, the first panelist. I'm gonna talk about biofats. I'm not sure whether this topic evokes your interest, but I hope you will enjoy my presentation. I will briefly introduce the biosensors first, then let's dive into our main topic, biofats. What is a sensors? A sensor is something that detects events or physical quantities. In a nutshell, a sensor is a system that has one input and one output, but they must have different physical forms. Then what is a biosensor? The only difference is that its input is related to bio things. Back in the years, diagnostic imaging was the most popular method. We can scan specific images like MRI or CT and check whether we get diseases or not. The limitation of those methods was obvious. It was hard to diagnose at an earlier stage due to its inconvenience. So nowadays, the paradigm has been shifted from diagnostic imaging to in vitro diagnostics using biosensors. The main advantages of in vitro diagnostics are its fastness and accuracy. So the possibility we can diagnose diseases can increase substantially. As seen here, there are so many combinations of input and output pairs. There are 4 types of input like DNA, antigen, enzyme reaction, chemicals. DNA needs complementary single-stranded pair on a surface, like this. And antigen needs an antibody, which targets specific antigen. We categorize biosensors with its output signal forms. If the output signal is electrical, then it is biofat. And there are many other forms like optical, mechanical, heat. A few years ago, many researchers utilized graphene, carbon nanotube, and nanowire due to its comparable size with these biomolecules. It means that photodetector, microcantilever, and thermistor were popular at that time. However, thanks to the CMOS downsizing, its size has decreased to just a few nanometer scale. And now we can make high throughput biosensor at a chip level with high developed semiconductor technologies. Here is the conventional FET structure. FET is an acronym that means field effect transistor. Induced gate voltage creates an electrical field, and it makes a channel which is a source to drain current path. We can consider it as a simple switch as well. If we induce a particular voltage level at the gate, then the channel will open and the current will flow like this. In BioFET, the gate electrode is replaced by electrolyte. It means biomolecules in electrolyte generate gate voltage, and it will create a channel and current flow. Next, let's briefly check BioFET's voltage current curve. In this graph, induced voltage creates the channel flowing current. And there are two main parameters in BioFET. The first one is subthreshold swing, and the second one is threshold voltage. Subthreshold swing is a slope in this graph. And high subthreshold swing guarantees high sensitivities. The threshold voltage is a level that BioFET is turned on. And these two parameters are closely related to each other. Now that we know what BioFET is, I'd like to explain in a specific situation. In this figure, there are biomolecules having negative polarity, negative charge. And antibodies are demobilized on the surface like this. If the binding events occur like this, it induces a negative gate voltage. And the channel will be closed and the drain current decreases like this. The BioFET fabrication process is quite similar to the conventional Mosfet fabrication. In this process, the SOI wafer is used. SOI wafer has a sandwich structure, which consists of Si substrate and Si O2 box and top Si. First, top Si is etched with remaining source and drain region. Next, cover the channel region like this, and then conduct surface, and then implant ions to the source and drain region like this. And then remove the photoresistor on the channel and conduct surface functionalization. Like make antibodies on the channel and deposit oxide and metal on the source and drain regions. And lastly, deposit passivation layer, which play roles of protection layer. After fab out the chip, drop electrolyte to use it in a lab. To demobilize antibodies on the surface as a receptor, we have to employ the surface treatment step. Typically, a hydroxy group presents on the silicon oxide surface like this. So after APTS modification, it displaces the hydroxy group to amino group, and then use GA to attach the carbonyl group on the amino group like this. Finally, the amino group in the antibodies react with the carbonyl group on the surface. This is bond. To prevent unwanted protein with attached to unbinding sites, we should use BSA to block the unbinding sites. Picture B and C are AFM images. It shows the roughness, and we can see the difference between before and after surface treatment. Lastly, I'd like to introduce the example of usage of BioFET in the field of NGS, next generation sequencing. The mechanism is pretty simple. when polymerized integrates a nucleotide like this, it releases hydrogen and pyrophosphate. As it releases hydrogen that has positive polarity, so we can simply sense it as an electrical signal. Here is more detail in picture A. Yeah, let's imagine we put guanine. If there is cytosine on the BiS side, then polymerase integrates guanine and it releases hydrogen. Then we can see the peak signal simultaneously. On the contrary, if there is thymine on the bead, then there will be no signal generation. That's all for my presentation. Thanks for your attention. Thank you.
Okay, thank you very much. Next up, Junte is going to talk about cantilever-based biosensors. Junte?
Okay. Hello, everyone. I'm Junte from BEE Lab of Professor Char Kim. Today, I'm going to introduce you about cantilever biosensors. So, cantilever biosensor is basically MEMS. So you can just think as the diving board I've shown at the first slide. So if someone is standing up on the diving board, the diving board will be bended like this. If some target materials are attached on cantilever biosensor, it will be bended and make some electrical signal. And when we use cantilever sensor as a biosensor, we should deposit the biomolecules on cantilever surface to sense target biomaterial like proteins or DNA. And for compare, the reference cantilever is needed also, which is not deposited with biomolecule. So this is the basic principle of cantilever sensors. The cantilever biosensors can be classified into 3 sensing modes. The first is static mode, and second is dynamic mode, and lastly is heat mode. So today I will deal with only static mode and dynamic mode. Static mode literally means the movement of cantilever is static. So we can just measure the bending degree of cantilever. And second, dynamic mode. When we use dynamic cantilever for sensing, we measure the resonant frequency of cantilever. To make dynamic cantilever vibrate, we set the environment in vacuum. And on the other hand, in order to make cantilever in static, we put this on laminar flow of fluid. So from now, I will introduce about 5 sensing mechanisms of cantilever biosensor. First is optical detection. So we shoot laser beam on cantilever and measure the light power on CCD, which convert the power of light to electrical signal. So we can sense diffraction or transmission induced by the diffraction of cantilever. So if the cantilever is bended, the power of light will be decreased. Second is piezoresistive method. So when there is a pressure on piezoresistive material, the resistance of this will be changed. So we make Wheatstone bridge with piezoresistive materials like this picture. So if there is a stress at one side, the resistance will be changed. You can see the delta R. So then we can measure the V out and current to sense bending degree and twist degree. So, and this is the microscope picture of piezoresistive cantilever. you cannot see where is piezoresistive material because it is wrapped by captivation. So we'll check it later in fabrication process. And next is capacitance method. So it's very simple too. So there is a conductive plate below cantilever sensors. So if cantilever is bended, the capacitance between cantilever and conductive plate will be increased. And next is Mosfet based method. So this is similar with piezoresistive material. If there is stress caused by cantilever deflection, the conductivity of Mosfet channel is changed and we can detect the current variation. And last is resonant frequency method. So I mentioned before, in dynamic mode, they use resonant frequency. So in this picture, they use some resistive method, but we can use all kind of method I've presented before, like optical, piezoresistive, capacitance, Mosfet method. The pros of dynamic mode is definitely accuracy because they use vacuum as medium. Then I will introduce about the fabrication process. Firstly, we make the wafer. So wafer from silicon ingot and make it oxidize. Then we deposit the polysilicon and etch to make this shape. And next we deposit the nitride which protect the bottom side of cantilever. And then we deposit doped polysilicon. So this doping materials makes polysilicon into piezoresistive materials and deposit nitride to protect top part of the cantilever and make electrode here for connect with line. And next deposit gold to make biomolecules can attach well on cantilever. And lastly, etching this side here chemically, and lastly the cover with biomolecule. So we've checked the whole simple fabrication process of piezoresistive cantilever biosensors, and this is the end of my presentation. Thank you for your listening.
Okay. Thank you very much, Shunta. So we're gonna have our last Hyun Kim, and he's gonna talk about fluorescent protein biosensors.
Can you hear my voice? Okay, thank you. Hello, I'm Hyun Kim, presenter 3 of Group 4. And here I will present about fluorescent protein biosensors. Fluorescent protein-based biosensors have less responsive time with high selectivity and low invasiveness. Thanks for these advantages, it becomes promising in clinical diagnostics, microscope-based analysis, and environmental monitoring and so on. Of course, there are barriers for fluorescent protein-based biosensors. In here, I like to talk about two famous fluorescent protein biosensors, FRET and BiFC. A fluorescent protein absorbs specific wavelengths of light and emits specific lights with longer wavelengths. FRET biosensors conceived this idea using two kinds of fluorescence proteins. When light excites donor fluorescence protein, its emitted light with specific wavelengths is observed by acceptor fluorescence protein. Again, acceptor fluorescence protein emits specific light. These kinds of energy transfer is called FRET, and these kinds of biosensors is called FRET-based biosensors. Since energy transferring efficiency is sensitive to the distance of fluorescence protein, FRET biosensors is used for monitoring protein-protein interactions, protein-DNA interactions, and protein conformational changes. These FRET-based biosensors have pros and cons. Since two interacting proteins should be brought in close proximity, maximum distance of FRET biosensors can be detected is FRET biosensors is low sensitive because small interacting protein. Also, it requires higher levels of protein expressions for energy transferring. However, FRET-based biosensors offer real-time monitoring and reversible fluorophore interaction. Thanks for these advantages, using FRET, rapid kinetics such as neuronal spikes can be monitored. Live figure shows one of example of a FRET-based biosensors application. To image neuronal spikes, CAMK2 activity is successfully detected by MECQ-M citrine and MECQ-M orange-2 FRET biosensors. Unlike FRET-based biosensors, BiFC-based biosensors uses dissected fragments of fluorescent proteins. Since each fragment loses fluorescent characteristics, they do not fluoresce themselves in this stage. Then each fragment is fused with other proteins, those interactions to be identified. If protein A and B interacts with each other to come closer enough, each fragment is recombined and fluoresces. These BiFC-based biosensors need extra long maturation time. And once fluorophore has been reconstructed, it is generally irreversible. However, it has not much tight operation distance like FRET, so that it can detect the distance over 7 nanometers. In addition, comparing to FRET, it offers high sensitivity because of its little background fluorescence. Lastly, it do not need higher levels of protein expressions, since BiFC do not have to transfer energy to other fluorescence proteins. Thanks to these advantages, BiFC can reveal subcellular localizations. Live figure shows interactions between movement protein of tobacco mosaic virus and CRT in tobacco leaves. CRT tagged with CEYFP and TMVMP tagged with NEYFP is interacted and localized to plasmodesmata. Using biopsy method, these subcellular localizations can be detected. In conclusion, fluorescent protein-based biosensors have many advantages, especially for systems which require fast, high selectivity, and less invasiveness. There are two famous fluorescence protein biosensors based on FRET and BiFC, and both have pros and cons. FRET-based biosensors offers real-time monitoring, irreversible fluorophore interactions. However, its maximum detectable distance is 10 nanometers. Also, it is relatively low sensitive and requires higher levels of protein expressions for energy transferring. On the other hand, BiFC-based biosensors requires long maturation time. And once fluorophore has been reconstructed, it is generally irreversible. While it has not much tight operation distance like FRET. In addition, comparing to FRET, it offers high sensitivity because of its little background fluorescence, and it do not need higher levels of protein expressions. Fluorescence protein biosensor is promising in many applications. Since there are various pros and cons, choosing proper fluorescence biosensors will be important. Thanks for the listening.
Thank you, Anka. That ends our presentation. So if you have any questions, you can either just unmute yourself or you can just post it in the Q&A.
If anybody have a question, please ask. Yep, there is a last question.
Yeah, from I'm sorry if I butchered your name, Dankil Suk. Says that there has been some introductions about the bioelectrical sensors, such as bioFETs. And what are the advantages of the bioelectrical sensors compared to the biochemical sensors?
Yeah, I will ask about this question. At first, we can fabricate it on chip level, so the size is very compact, and we can programmable manufacturers like sensitivities or the minimum detection limit. So I think the programmability is very great pros for BioFast rather than using biochemical sensors.
Is there also a question? If there is no question, please open or moderator finish this seminar.
Okay, that was the seminar about biosensors, and I thank our panelists and presenters, and thank you everybody for listening to us. That'll be the end of it.
Yeah, bye.